Cyanagen is pleased to present the StarBeads Viral DNA RNA Extraction Kit in Prefilled Plate Format. The kit is validated for COVID-19 diagnostics and CE-IVD certified. The purification method is based on magnetic bead separation technology, and all individual components and reagents, including the magnetic beads, are manufactured at the Cyanagen site. The extraction of nucleic acids employing commercially available kits in bottle format is often time-consuming and laborious. The main disadvantage of these bottle format kits is that the end user is responsible for separately acquiring and maintaining sufficient stock of expensive plastic consumables. A second major disadvantage is the technician hands-on time to prepare the individual reagents, often to be stored at 3 different temperatures and usually supplied in a not-ready-to-use form. Therefore, they must first be diluted and then dispensed manually in 4 96-well plates for 96 test extractors. Only then the technician will be able to add the analytical sample. This entire process requires 30 minutes at least and is susceptible to human error. By developing the StarBeads Extraction Kit in prefilled plate format, Cyanagen aims to speed up the preparatory phase to extract nucleic acids, thereby saving precious time and hence the opportunity to process much more COVID-19 samples reliably. The StarBeads Viral DNA RNA Extraction Kit in prefilled plate format is available in multiple configurations. Prefilled 96-well plates for 96 samples consists of a viral sample plate, a washing 1 plate, a washing 2 plate, an elution plate, and a tip comb plate. The kit is also available in prefilled plate format for 32 samples, consisting of a viral extraction plate and a rod's tip. Employing the StarBeads Viral DNA RNA Extraction Kit in prefilled plate format, the preparatory phase for the extraction of nucleic acids is significantly faster and eliminates potential human error. The technician merely has to gently rotate each plate 5 times and then spin down for 30 seconds. Add the analytical sample. Hands-on time is reduced to only 5 minutes than the required 30 minutes when using bottle format kits. Further, the kit is compatible with different matrices from which the genetic material can be extracted, such as serum, swabs, saliva, and other bodily fluids. In addition to significantly speeding up the preparatory phase for the extraction of nucleic acids, Cyanagen has developed efficient, reliable, and fast extraction protocols and scripts. Only 18 and 22 minutes are required for the extraction of 96 samples and 32 samples, respectively. The extracted genetic material is of high purity. It is immediately ready for use in any downstream application, such as RT-PCR, quantitative RT-PCR, Sanger sequencing, NGS, PCR, and other enzymatic reactions. StarBeads Viral DNA RNA Extraction Kit is highly flexible. Protocols and scripts are available for the most common automated extractors. For more information, visit cyanagen.com/StarBeads Viral DNA RNA Extraction Kit.